Nowadays, any disease you have, it's attributed to either the microbiome or inflammation. So we should try and understand it. So, okay, that's not good. Okay. So inflammation you can think of in two ways. One is acute inflammation and the other is chronic. Acute inflammation is really a protective immune response that normally occurs after an injury such as a physical cut or a sprain or an infection. And once the problem is resolved, the acute inflammation ends. But if the problem persists, then you get what is called chronic inflammation. And many of the diseases that you hear about in HIV and also in aging are really considered an outcome of chronic inflammation, such as the ones listed here. And including also things like metabolic syndrome, dementia, and frailty that you heard about earlier this morning. So let's talk about HIV and inflammation now. Once the virus is inoculated into the host, there's the burst of viremia, which then settles down into a set point. And the body reacts by an inflammatory response, and you get a whole bunch of cytokines. Cytokines are proteins that are liberated from activated immune cells. And they are meant to be, again, a protective response. But in usual infections, if there is inflammation, you would expect it to cool off when the infection is resolved. But HIV persists, and because of the chronic phase of the disease, you have continued inflammation. And that's what we're going to kind of discuss. So, what are the common questions about inflammation in HIV disease? What causes it, and why is it bad? What causes it? Some of it was discussed this morning by Dr. Letendre. And so one major cause is that once infection occurs with HIV, very early on in the course of infection, there is damage to the gut. The virus multiplies in the lymphoid cells, in the gastrointestinal tract, and intestinal integrity is compromised very early, and that leads subsequently during the chronic phase of inflammation of HIV, gut microbial translocation, and that's considered a major cause of inflammation. Then activation of co-infections such as CMV and EBV, also as that you heard about this morning, lead to inflammation. And with HIV, there is immune dysregulation. There's HIV persistence. The virus is not eradicated. It settles into latent reservoirs. And then, of course, we've heard a lot about the microbiome in HIV. It's altered, and that is also one of the factors in leading to inflammation. And why is it bad? Because it leads to all those diseases. It's suspected that this is one of the causes for it, the end organ diseases. It also leads to fibrosis in lymph nodes. It leads to immune senescence, and it leads to impaired immune function, and viral reservoirs are activated occasionally because of persistent inflammation. So I'm going to discuss the end organ disease, which is mainly cardiovascular disease, and impaired immune function. And so in today's discussion, we're going to talk about two studies related to inflammation and immune activation. One is immunity in HIV and aging, and this is in a US cohort on ART that was virally suppressed. And second is HIV and cardiovascular disease in the Chennai cohort, including patients not on ART as well as patients on ART. So first, let's talk about HIV and aging, and I'm going to only talk about immunity in the context of aging. and not really the other end organ diseases that were discussed by Dr. Letendre. So what about immunity? We know that with biologic natural aging, the immune system declines and leads to increased incidence of infection. Also in HIV, there is increased incidence of infections. And it's still debated if in treated HIV, there is immune senescence that is hastened and also known as accelerated aging. And the goals of this study were to evaluate immunologic changes associated first with healthy aging to understand better what occurs in healthy aging, and second, to evaluate impact of controlled HIV infection on immunity in young and old, both common features as well as differences. For this study, we enrolled people who were HIV positive and negative in 3 age groups, what we call the young, 18 to 39, middle age, 40 to 59, and old, greater than 60. And we enrolled about 140 people in each HIV positive and HIV negative groups. All HIV patients were on ART. And we needed an in vivo mechanism to understand whether the immune system was functioning. And for that, we used the trivalent influenza vaccine, which is given every year to people in the United States. And the response to the vaccine was used as a probe of immune system function. And there were 4 questions that we wanted to address. Is the immune system of old, healthy people worse than that of young, healthy people? And second, does HIV lead to accelerated aging? Means, do these young people become like old people if they are infected with HIV? Third, if old people get HIV, or we see HIV in the aging population, is it worse in them than it is in HIV-negative people? And fourth, and most important, can baseline immune parameters predict the response to vaccine? So we did a bunch of immunologic investigations, and you can imagine we use peripheral blood for most of these studies. And in our peripheral blood, the white cells are made up of many subsets of immune cells, the T cells, the B cells, monocytes. And so you can investigate these very simply by looking at the surface molecules on the cells, which we call immune markers. And you do that by flow cytometry technology. And we also looked at the plasma of these patients and looked at these inflammatory cytokines and microbial translocation products. And then we correlated it with the outcomes of age, HIV status, and serologic vaccine response. So the first, I'm going to tell you whether how the vaccine response occurred in these patients. And this study was led by Suresh. He came from Kerala, first as a postdoc, and now as a faculty member. And on the right side, you see the responses to the vaccine. First is the response to one component of the vaccine, the B antigen, and the bottom are all 3 antigens. It's a trivalent vaccine. And the dotted lines indicate HIV negative. The solid lines are HIV positive. Reds are what? Reds are young, and old, and blue are old. And so you can see the first question was, is the immune system of old healthy people worse than that of young? And so you can see that young negative is up here, and old negative is down here. So you can really see the response is much better. These are different time points. 0 is baseline, T1 is 1 week, T2 is the time when we really measure the serology, and that is at 4 weeks. And then to see the decay, we looked at 6 months. And so it's clear that the young were much better. And then the second point was, does HIV lead to accelerated aging? Can you see it in young HIV infected? And you can see the young HIV positive is very similar to the old HIV positive and negative. And so, yes, there is some kind of accelerated aging. And the third and the fourth points we could not address by this serological assays. And then we looked at the different subsets that I talked to you about. And this study of B cells, we found this was led by Stefano. He came from Rome, as he was very interested in B cells, so he addressed the B cells in this project. And he found, as you can see in the title, that immune senescence of B cells is most prominent in young age. Again, that the young HIV positive are very similar to old HIV negative. And you can see that in a 1-week response, antibody response of HIV positive young individuals who are very similar to old negative and old positive, and very different from the young negative. And then the third project is really related to baseline markers of immunity, of inflammation, how it affects the immune system. And this work was done by Leslie. She came as a technician, then did her PhD, and then came back as a postdoc. And the important points are underlined. And basically, the major points are first, that activated CD4 T cells, which are characterized with these markers, CD38 and HLA-D4, DR, which are assessed by flow cytometry, are much, much higher in HIV positive than in HIV negative, but they show no relationship to age. So it's really a characteristic of HIV infection and not something that is a feature of biologic aging. And then we looked at cytokines, and the point of this slide is to tell you that the green box indicates cytokines that were only affected in HIV negative old people, and the red box shows you cytokines that were affected in HIV negative and positive people with aging. So the correlation with aging is really different in HIV positive and negative with some similarities, whereas this IL-8 was only seen in HIV positive. And this slide shows you comparison between HIV and healthy controls. And you can see the differences between HIV and healthy control maximally occur in the young age group and are fewer as aging advances. Again, young HIV show most differences from healthy controls. And finally, here we looked at modeling using what we call lasso technique. Lee Penn, who's our statistician, did this work, and she took all of the markers that we looked at and did these plots. And you can see that as the red triangles are responders, the blue are non-responders, and you can segregate responders and non-responders. And the baseline immune characteristics predicted influenza responders and non-responders, but the immune signature was quite different with some overlap, telling us that although, yes, you do get responders and non-responders, the pathway to reaching that is maybe different in HIV positive and negative. So in conclusion of this part of the talk, you can see that HIV-related immune senescence was clearly evident in young HIV-positive patients. It was characterized by a core signature of immune activation of CD4 T cells, but was distinct from biologic aging. And biomarkers predicted for antibody response to vaccination were not identical between HIV and healthy controls. There were many more negative predictors in HIV than in healthy controls. And so we hypothesized that the immune activation and inflammation underlie immune dysfunction resulting in impaired vaccine response, but components of factors that lead to altered immunity are most likely different from those involved in comorbid health events like the cardiovascular and the other aging-associated events. So the next component of this presentation is HIV and cardiovascular disease. And this study was done mainly in Chennai, and there was the brainchild of Dr. Kumar. And the two people who took the lead in this were Kausalya and Dr. Sarvanan, and together with other members of the VIRG care team. And so the question here was, what is the relationship of CD4 count on cardiac function, as well as measures of immune activation and inflammation? How do patients on ART differ from treatment-naive viremic patients? And which immune measures correlate with cardiac function, particularly which measures of inflammation? And here is the study design. So we had 3 groups, Group 1, Group 2, and Group 3. Group 1 was the ART naive, untreated, and there were 3 subgroups, 1A, 1B, and 1C, with different CD4 counts. Group 2 was on ART for at least 1 year, so they were really reconstituted. But the important point is that they all started their treatment at the same nadirs as the Group 1. And then Group 3 was the HIV non-infected. And you can see that despite the fact that they were immune reconstituted, the Group 2 was still different from HIV negative healthy controls in terms of CD4 count. So first, let's see what the cardiac status was. The cardiac functions were assessed using this pulse wave equipment that has been used in the ACTG trials also. And the main functions were ejection time, stroke volume, and cardiac output, as well as arterial measures of resistance and elasticity. And this slide shows you that Group 1 has the most impairment in cardiac function, and that's the Group 1 in red with the lowest, which was untreated, compared to Group 2 and Group 3 in all of the major cardiac functions. And this is a heat map just showing the subgroups, 1A, 1B, 1C. And this is really to illustrate that most of the defects of cardiac abnormalities were seen in Group 1A, with some abnormalities in Groups 1B and 1C with the higher CD4 counts, and some residual abnormalities in Group 2. So next we went to look at what was the immunologic basis. Could we correlate these? And here we did 4 parts, the study in 4 parts. We looked at markers of immune activation. We looked at markers of plasma, biomarkers of inflammation and microbial translocation. And then we also looked at checkpoint inhibitors and monocytes and correlated them with cardiovascular function. And this shows you that first, looking at T-cell immune activation markers, the immune activation is highest in the treatment-naive group, and maximally in Group 1A as compared to the other groups. And immune senescence, which was studied by CD57 and 28 negative cells, was mostly seen in CD8 subsets, and again highest in Group 1 and highest in the Group 1A, also particularly high in Group 2 subsets. And this shows you the correlation with immune of these CD4 and CD8 cells immune activation with the cardiac functions. And you can see that the immune activation was correlated with reduced cardiac function, as shown in blue, compared to healthy controls. And immune senescence was not correlated, by the way. And then we looked at plasma biomarkers, and again you can see mostly the red is bad, and that's what you have to remember. And you can see that it's the Group 1 that has the most inflammatory biomarkers and some residual inflammation and microbial translocation markers also in Group 2. And this is the heat map that is showing you that cardiac dysfunction is associated with multiple plasma cytokines and LPS, and increased inflammatory cytokines are a result mainly of immune activation. And lastly, we wanted to look at monocytes and checkpoint inhibitors. And checkpoint inhibitors are molecules that are present on T cells. And when T cells get activated, they have to interact with the receptors on monocytes. And these receptors are required for T cells to proliferate, as well as receptors are required for the response to come down. And PD1 and CTLA4 act at a systemic level, and whereas TIM3, LAG3, and TIGIT are important markers which act at the tissue level. And they are expressed in tissues, and their anatomical specifications in different tissues express different of these checkpoint inhibitors. And you can see that in the T cell subsets, there was higher PD1 and LAG3 and CD4 T cells in group 1. And again, shown in a heat map, these markers are mostly in group 1A, but also persist a little bit in the other groups. And this is showing correlation of these markers with cardiac function. And red line is, again, group 1. And you can see it going down. And both when the CD4 cells express LAG3, or they co-express LAG3 and PD1. And this is a heat map, again, showing that LAG3 and PD1 expressing CD4 T cells are correlated with cardiac function impaired cardiac function in group 1. And this is due to increased immune activation. And finally, we come to monocytes. And monocytes, we know that there is evidence that monocytes are important players in inflammation. They're implicated in atherosclerosis and plaque formation. And there are three major circulating subsets of monocytes: inflammatory, classical, and non-classical. And these monocytes can be distinguished by markers on cell surface, mainly CD14 and CD16, again by flow cytometry. And their functions are quite different. Classical, which constitute the maximum numbers, are important for phagocytosis. Inflammatory are, as their name implies, pro-inflammatory, and non-classical are patrolling. And all of these markers, all of these monocyte subsets have CD11B, which is another marker. It's an integrin marker, integrin molecule on the cells, which can be expressed high or low. And this has a very important function in these cells. You can identify all of these subsets by flow cytometry quite easily. They're quite distinct: classical, inflammatory, and non-classical, and based on the markers that I told you. And Varghese, who was a postdoc in my lab, did this in the aging study and found that CD11B high expressing inflammatory monocytes were really detrimental for the vaccine response. So we looked at these same markers in the Chennai cohort, and we found that there was, first of all, no difference in the subsets of the monocytes, the classical, inflammatory, or non-classical, in either the untreated or the treated or healthy controls. They were all the same. But when we looked at the CD11B high expression, it was quite clear that CD group 1 had the higher frequencies of CD11B in all the monocyte subsets, particularly in the inflammatory and the classical monocytes. And this is a heat map showing you the summary of the monocyte subsets in the Chennai cohort. And so CD11B expression of inflammatory and non-classical monocytes was inversely correlated with cardiovascular disease. Means the cardiovascular disease was much worse in the presence of cells that expressed the CD11B high monocytes. And you can see that it's mainly in group 1, with just a little bit of that effect still persisting in group 2 when you compare it to healthy controls. And so what is the mechanism of activation of monocytes? We think it's through the receptors on the cells, the TLR signaling. And as we talked about microbial translocation, the HIV viruses and bacterial products can all activate these TLR receptors on the monocytes, which are outside and inside the cell, and lead to activation and lead to inflammatory cytokine production. So in summary, for the Chennai cohort, untreated patients have significant immune dysfunction, immune activation, inflammation, increased checkpoint molecules, and increased CD11b high monocytes. This was most prominent in group 1A with CD4 less than 200. Patients starting ART at any nadir showed immune recovery, but this was less in those who started at lower nadir CD4. Some immune dysfunction, such as soluble CD14 and immune activation, is evident even if ART was started at higher CD4. And cardiac dysfunction correlated with immune activation of CD4 T cells, increased inflammatory cytokines, increased checkpoint inhibitors, LAG3 and PD1, and increased CD11b high monocytes. Maximum defects were in group 1. So coming back to the common questions about inflammation, we discussed what causes it and why is it bad, but what determines which organ system is affected. And we think specificity is at the tissue level. It could be because of receptors for inflammatory cytokines. It could be because of ligands for checkpoint inhibitor molecules such as LAG3 and PD1, especially in the heart, where we saw these playing a role. And it could be because of migration of effector cells and inflammatory monocytes. We don't know how the signaling can affect the damage to the end organ, but it's really important to understand this. We know that early treatment with ART can probably reduce inflammation, but there need to be strategies considered for people who have ongoing cardiac disease and inflammatory basis for it, to really think of strategies to intervene so that we can overcome this problem. And so it's really a jigsaw puzzle at this point. We really have to figure out how all these things fit, and especially co-expression of co-inhibitory molecules could play a very major role. And at this point, I'd like to acknowledge people in my lab, and especially in YRG Care Chennai, where most of the work for the second study was done. And thanks to all the study participants and the study team personnel who helped in this, and to our CFAR, YRG Care, and NIH for the funding of the Indo-US project that helped us to do this study. Thank you.